Good morning and welcome. This is the second inventor webinar that we're broadcasting from Stanford University. My name is Scott Elrod and I'm Associate Director in the Office of Technology Licensing. And I'm pleased today to introduce two inventors who collaborated to create this invention. First, Professor Hanli Ji. Professor Ji is an Associate Professor of Medicine and Oncology. He's also the Senior Associate Director of the Genome Technology Center here at Stanford. He has his MD from Johns Hopkins University. And presenting today will be his collaborator, Dr. Billy Lau. Billy is a postdoctoral scholar in the School of Medicine, and he has his PhD in engineering science from Harvard University. A few details about the webinar. If questions come to you during the course of the webinar, we'd invite you to submit those now. We'll roughly have 20 minutes of presentation and 10 minutes of Q&A with people in the audience. If you look around the perimeter of your Zoom screen, you'll see that there are two functions that might be used for questions. One is called Q&A and the other is called chat. We'd like to urge you to use the chat function since questions then come in anonymously. And so we'll be looking in the chat window primarily. So please use that. And you can enter questions at any time. So with that, maybe I'd ask you, Billy, just to describe briefly the subject of the broad topic of research in your group, and then after that we'll jump to the slides and take a more in-depth look at this invention.
Yeah, so Hanli's group is jointly appointed between the Division of Oncology and the Department of Biochemistry at Stanford University, and it's really like an interdisciplinary research group where we develop sequencing technologies as well as have an extensive tumor biology program to understand patient-derived tumor malignancies with the context of understanding how they grow, maybe what the treatment options are, and understanding what resistance mechanisms may exist.
Great. All right, with that, I think we'll switch over to the slides and spend about maybe 20 minutes going through slides. And again, we'll open it up to Q&A from people who are in the audience.
Great. Okay, so so today I'm going to be talking to you about some of the results from a recent research project in the sequencing technology side of Hanli Ji's lab. Today I'll be talking to you about a new method to perform molecular barcoding for next generation sequencing. So as I said, with this barcoding technology, I'll be talking to you briefly about what molecular counting is in next generation sequencing, and also go over briefly what some problems that existing technologies have currently. And to address these concerns, we'll be talking about some recent results. We address these recent results using a technology that we developed in-house called Exponentially Expanded Barcodes. So as I said before, in the sequencing technology side, we seek to understand characteristics about tumors themselves, namely basically what mutations may drive the cancer, and maybe see if we can find some biomarkers regarding treatment options, as well as maybe possible biomarkers that would lead to downstream resistance to treatments. All of these fall under the realm of quantification of some kind of nucleic acid molecule. So for instance, in the DNA realm, we would look at things like tumor purity or tumor heterogeneity, which means we would study how much of a tumor is present in a tissue biopsy, and tumor heterogeneity basically addresses how different are individual cells in the tumor from each other. And this has important implications in studying whether patient's tumor will later have some kind of resistance to targeted therapies or even just general chemotherapy. So more generally, this question on the DNA side, we're looking to count how many mutations or what kind of genomic aberrations are there at a particular genomic site. On the RNA level, we're looking to quantify what the particular expression is of some kind of gene of interest. This could be like an oncogene or a tumor suppressor. And this is important because the expressions of these oncogenes and tumor suppressors are directly correlated to malignancy themselves, because ultimately DNA will eventually be transcribed into RNA, and then it will eventually turn to proteins, which are the ultimate phenotype of cancer. And in this, more generally, we'll be looking at how many reads in the NGS assay correspond to this gene. So all of this in general revolves around the accurate counting of these of molecules, whether it be from DNA and RNA. So the existing technologies that exist basically count NGS reads, but the problem with this approach is that unique molecules from, let's say, from a tissue sample or from genome DNA or anything that's purified, they can be represented by more than one read because of some kind of PCR duplication. The one way to address this problem is to use these things called molecular barcodes, or otherwise in literature are known as unique molecular identifiers, or UMIs. These can tag DNA fragments before PCR, and what happens is that during the PCR process, these tag sequences get copied as well. So by tracing the individual identities of the tags, you can tell what is the extent of PCR duplication and basically be able to resolve what is duplicated and what is not. So you can tabulate these abundances and you can identify them pretty easily in your sequence data. All you would do in your alignment files or in your FASTQ files is to say, give me all the reads with particular barcodes, and then you would see what's duplicated and what's not. What's commonly used is stretches of random nucleotides. So in this case, what happens is that during the ligation process or whatever process you use to create library fragments, you have a stretch of fixed length of basically a bunch of ends. And the total diversity of these ends is larger than the number of DNA fragments that are in your reaction. So what happens is that each individual DNA fragment gets tagged with something that's unique that nobody else has. And through a PCR process, by counting these molecules, you can basically tell what is a PCR duplicate and what's not. The problem with this approach is that PCR, I mean, the previous approach, I think, it's very clever and lots of people use it, but there are a few shortcomings. So PCR sequencing assays have inherent error rates, depending on the sequencing reaction you have or the PCR reaction or any kind of assay you have to generate these libraries. They have some sort of error rate, which means some sort of probability of misincorporation of a certain base. What this means is that the specific barcode identities may not be perfectly measured. So what I've shown you on the top right is basically one barcode sequence as a top line and a series of mutations that might happen in this barcode with different sequencing reads. Some of them are single nucleotides, some of them are double substitutions. The problem is that because random barcodes are, they're random, there's no prior knowledge about what these barcodes are, so that it's actually pretty difficult to group together these this barcode and also the errors and say what the original barcode was. Some methods do exist, but they're mainly very heuristic or very ad hoc. There's no really real systematic way to do this with extremely high accuracy. So one solution that has come out is to use pre-designed barcodes. What that means is that you have to individually synthesize every molecular barcode. What I've shown on the bottom right is 2 example barcodes that are bolded and a series of substitutions that may occur in the barcode sequence. So because we pre-designed these barcodes, it's pretty easy to detect the errors because all we have to do is in your sequence reads match which of these barcodes is like basically the closest match based on edit distance or some other metrics. The problem with this approach is that this is really not scalable to studies with lots and lots of molecules in your sequencing reaction because there's such a high complexity. You would have to be able to synthesize every single molecular barcode by individually on either in plate-based form or what have you. And this actually gets pretty expensive to be able to synthesize every oligonucleotide barcode. So to address these concerns, we have devised a method to exponentially generate large combinations of pre-designed barcode sequences. And we call this exponentially expanded barcodes. So what we have on the bottom right is like an example structure of the scaffold that has these exponentially expanded barcodes, or so-called EXBs. What it is that it is a series of barcode sequences that are surrounded by scaffold sequences that are assembled together. Flanking the sequence is also a random nucleotide sequence shown in green, which I'll get into later. But in each individual red position, you have 64 different possibilities. And if you look at the bottom right panel, it's basically showing us a summary of the pairwise edit distance between individual barcode sequences. And what I've shown is that the vast majority of sequences have an edit distance. These are 6 base pair barcodes. That the distances between each individual barcode is extremely high, so that they're very distinct from each other. I would also like to say that the study is already published in BMC Genomics last year, if anybody wants to take a look. So more details about this adapter sequence. So these EXBs combine results from digital signal transmission theory, where we generate redundancies in a barcode sequence so that they are very distinct from each other with high edit distance. Also with molecular biology to create very robust barcodes. As I said before, these are combinatorially generated in a pool. This is a pooled assembly of barcodes generated by this generator matrix, which is that little box of numbers there. And what this is that it allows for very high throughput generation of very distinct barcodes using a known mathematical formula. Sorry, I need to switch this to that. Oh, it's not working. So these exponentially expanded barcodes are amenable to different types of library preparation assays. So what we can use different types of methods to generate DNA libraries, but the one that we've shown in the paper is using TN5 transposase. For those of you who don't know what TN5 is, it's commonly known as the Nextera assay that's offered by Illumina. It's basically the same, using the same type of enzyme where we have a transposase molecule that's loaded with this adapter, and it can tagment inside cDNA inserts or genomic DNA or what have you. And from this, it creates inline molecular barcodes that we can read the molecular barcode sequence. And afterward, we can read these inline barcode sequences and group together the barcodes into individual EXB read groups, as shown in the bottom left. So we have examples of different numbers of each barcode sequence, and these are grouped together, and these are subsequently deduplicated, and then you can be aligned and counted as similar to random molecular barcode workflows. So this workflow is actually very familiar. But for the deduplication part, we have a new type of workflow. I'm not going to explain this in much detail because it's kind of complicated, but it's listed in the publication. But we have essentially a method to very quickly determine whether there are errors in the barcode sequences using results from signal transmission theory, where we have that matrix that I showed you in a couple of slides ago. We can actually play around with that and be able to figure out directly whether there are any errors in the sequence themselves using a very elegant mathematical trick. So from this, we can detect errors in the barcode sequences very systematically, as opposed to using heuristic approaches with randomer barcodes. So as a proof of concept that we showed in the paper, we applied this EXBs loaded with transposases to RNA-Seq. As a demonstration, we applied this to MAQC RNA standards. So for those of you who are really into doing RNA-Seq studies, the MAQC is a consortium of groups in the US, which sought to understand the reproducibility and the accuracy of quantifying transcriptome or RNA molecules. And these samples are basically reference brain and human reference RNAs that we reverse transcribe with cDNA first, and then we tagment with EXBs. And this type of data actually has lots of public resources in terms of results. So it provides a nice ground truth for us to measure the accuracy of our data. So we first, what we did is that when we tagmented this type of material, we checked to see what the duplication rate would be with lower and lower amounts of cDNA input. So when we see what we see is that on the bottom left is that as we lowered the amount of cDNA, we see an according increase in the number of duplicates per EXB group. So we can see that this assay is working that at 100 picograms, we see a substantial number of reads that are duplicates, similar to results that you would typically see with random molecular barcodes. What we also see is that using this assay with EXBs and TM5 transposase, that we can recapitulate the quantification performance as shown on the bottom right panel, that are with results that are previously known with our qPCR. What we can also see is that there's not a significant amount of technical bias in these assays. What I've shown you here in this panel is a pairwise correlation matrix between replicates of two different sample types. So there's 3 replicates of the brain sample and 3 replicates of the universal human reference RNA. And from here, basically the correlation coefficients of the brain samples and the correlation coefficient of the human reference samples mutually segregate apart from each other. What this means is that there is pretty good technical reproducibility and not such amount of technical bias such that these samples would blur apart from each other, so that these can be distinguished from each other. What we also see is that when we compare results from here with random molecular barcodes, we see that our barcodes can pick up a lot of errors that were otherwise undetected. So what I mentioned before is that in the sequencing adapter, there is a stretch of random nucleotides in front of it. It's basically that green thing in the adapter basically says N6. There's basically 6 random ends in the sequence on both read 1 and read 2. What we can do now is that by grouping together all the reads associated with the EXB barcodes, we can then look at what are for these reads in the EXB read groups, we can now look at the identities of these randomer sequences and then count how many errors there are, and also look at the quantification as if we were only looking at the randomer sequences themselves. And what we see on the bottom right is that as the regroup duplication rate goes up, the probability that actually one of these, some of the reads with these randomer sequences would have an error, it goes up actually pretty high, such that when you get a duplication rate of about, let's say 50, you have actually about 50% of, you have 50% probability of actually an error occurring. So when you sum this up over all of the possible EXB regroups, it's actually a substantial number of reads would have some sort of error. To further quantify this, we can look at the number of distinct random groups, randomer groups in each EXB read group on the bottom left panel. And we can see that what would happen if we were to use randomer barcodes is that we would create lots of false positive read groups, which would interfere with measuring gene expression of a read. They would basically be saying that there's an extra molecule in there when there actually was not one. And on the right panel, what we can see is that to correct these errors to correct these randomer errors would actually be very difficult. What I've shown on the right-hand side is the edit distance of these randomer sequences that have errors in them, and to check what is the actual distance between these barcodes. And what we see is that, like, while the average is actually quite modest, there's actually a very high standard deviation in these sequences, such that so some of them, the average has 1 edit distance between randomer sequences, which is not bad, but there are actually a very substantial number of sequences that have 2 or 3 errors. And what that means is that this is actually very, very difficult to correct with downstream heuristic metrics, whereas these were actually all picked up by the EXB sequences. So to measure the impact of these types of errors, what we've done is to look at the relative difference in gene expression between the EXB and random molecular barcoded quantification strategies. And what we see is that at low input amounts, we have, like, on the order of thousands of genes are incorrectly counted by, orders of magnitude. Like, so there's like over 10-fold or over 20-fold basically a difference between the 2 different methods. And this effect gets substantially worse as the input as the input amount decreases. So on the left side, we see, at 10 nanograms, which is actually quite a high amount of input cDNA, we see, there's some kind of difference, but it's not that substantial. But as soon as you move to 100 picograms of input cDNA, you see that this change is actually quite substantial. So this would really have impact on applications where molecular barcodes are actually really important. People typically use molecular barcodes in applications where you have very, very tiny amounts of input. What we've shown is that using EXBs helps you remove a lot of the errors associated with quantification. So finally, I'd like to wrap up this brief introduction to our technology by saying that there are other applications for EXBs besides RNA-Seq. So for one, you can use it to correct sequencing errors. You can apply this to single-cell sequencing, and more intriguingly, you can actually use this molecular barcoding technology for other sequencing platforms. So for those of you who are not convinced that this is a application for Illumina sequencing because Illumina's errors are quite modest, this would be actually a perfect application for PacBio sequencing or for Oxford nanopore sequencing technologies, where the error rates are at least like 20 times higher than Illumina platforms. So with that, I'd like to thank you for your attention. There's a link here if anybody would like to further explore this technology, and also some contact information if you're interested in talking with us about licensing.
Great. Well, thank you very much, Billy. So we'll just leave this slide up for one more moment. There's a Stanford reference number. We have a TechFinder website where you can actually look at more details of this invention as well as many other inventions. Jimena Aries is the licensing associate responsible for this technology, and you can contact her at the email shown here. And I'll come back to this slide at the very end for anybody who didn't get all the details down. But at this point in time, we'd like to open it up for questions, and again, invite you to use the chat function. If you don't see the chat function on most of these user interface controls, there's like 3 dots in the control bar, and you can get to the chat function that way. And if you can't find that and you need to, please use the Q&A function. That'll also work. So with that, we're opening it up for questions, and invite those questions from the audience. So I have one question here coming in. What is the lowest input cDNA you've tested with the EXBs?
So in the study in the BMC Genomics paper, we've shown only part of our results. We tested it on bulk cDNAs, but we've also done this on single cells, and it works there. Obviously, this is for a transcriptome of single cells.
Okay, thank you. Another question is, how easy is it to implement this?
So the nice thing about this technology is that when we created these adapter sequences, we use off-the-shelf molecular biology assembly protocols. So for people who are interested, we're using Gibson assembly to do this scaffolded assembly. I guess for people who are well-versed in molecular biology, this is actually quite trivial to actually create these adapters, and also to load the TF5 transposase. All of these are generally commercially available.
Thank you. Another question is around the cost. Can you estimate the cost for this process?
So my guess is that, the nice thing again about this technology is that because we're creating exponentially large numbers of barcodes from a smaller pool of oligonucleotides. In our sequencing assay, we actually only use about 192 oligonucleotides to generate an exponentially large amount. So in this case, we generated about 260,000 unique barcodes that are in Paradigm, create 60-something billion. But in the beginning, it's only 192 oligonucleotides to start. So that cost is actually quite small. It's less than $1,000. And all the other enzymes that are required to do the assembly and the TF5 transposase, that all is pretty inexpensive to purchase.
Okay, got another question here. Do you have any plans to test on other platforms, such as those that you mentioned, PacBio or Oxford nanopore?
Yeah, the answer is yes.
And when is that likely to occur?
Soon. Now, essentially. This is something we're actively doing, but it's an active research area, so I don't want to give away too many things.
Okay, great. So I'd ask, are there any other questions from our audience? We have a few more minutes, about 3 minutes until the end of the half-hour slot. Yeah, we just chatted out also the name of the Stanford docket as well as the TechFinder link. So if you go into your chat window, you can actually copy that. Here's a question. How many bases of sequencing read are devoted to the EXBs?
Yeah, so in the first implementation of this study, this is the one that's in the BMC Genomics one. It's actually quite a long sequence that I admit. It's actually about 70 bases of sequencing reads for the actual barcode sequence. And so you, in a typical 151 by 151 assay, you get about 70 bases of insert, 70 bases of barcode. So as sequencing technology improves, so nowadays you can get easily 250 bases for individual read. We're also working on methods, and also this is, I think, in the actual patent application itself. The actual scaffold sequence can be shortened quite a bit. And in the BMC Genomics paper, we've basically used the longest possible one to basically have more robustness, but you can actually shorten the sequence so that you don't need 71 bases of sequencing for the actual barcode sequence. We have actually have some results that are not disclosed here where we can actually bring that scaffold sequence actually to nothing.
Another question, how long do the barcodes end up being?
The individual barcodes or the total one?
Maybe you can answer both.
Yeah, so that's the same question as before, right? So,
okay.
So they could be, so in the implementation of the BMC Genomics paper, they're actually up to 70 base pairs, but this can be shortened down to basically just 18 bases as we're working right now internally. You can actually remove this scaffold sequence using some clever tricks. Yeah. Good.
Another one, do you think there'll be any impact on the EXBs with highly methylated DNA samples?
It depends on how you're generating the library. So right now we're using a TN5 transposase for insertion that has its own technical biases for methylated DNA, but also in the same time, this adapter sequence is highly flexible for different types of library preparation. So we in previous iterations of this technology, we can actually use just plain old DNA ligation to generate sequencing libraries. Which means it's completely amenable to things like bisulfite treatment and what have you.
All right, great. We have about 2 minutes left, so if there are any other questions, oh, here comes one. When you compare to UMIs, how many bases were the UMIs?
So
how will it change if the UMIs are longer?
Okay, yeah, that's a really good question. So the UMIs in total from read 1 and read 2 were a total of 12 bases, which gives you about, I think, like 16 million possibilities in terms of total diversity. The interesting thing is that people always talk about if you increase the number of UMIs, you increase the quantification performance. But what is talked about in the paper is that when you increase the amount of UMIs, because these UMIs are random, there's actually a trade-off, because the more random bases you have, the higher probability that polymerase will come in and actually create an error inside this random sequence, so that so you're basically, in some ways, making it worse by having a longer sequence itself, because as the read goes on, you have lower sequencing quality, and as the read goes on, the polymerase during PCR will create more and more errors cumulatively over the entire barcode sequence.
So I think we'll take one more question, and then at that point, I'll put the last slide up so people can see the contact information again if you didn't capture that before. This question is, how much better are the gene expression measurements with UMIs than without?
Well, it actually depends on how much input you have. So if you, with both UMIs and EXBs, if you have lower and lower amounts of input material, then the importance of using some type of molecular barcode is going to go up drastically. So in studies where you use a lot of material, if you luckily have a lot of tissue, or if you're doing from cell lines and you just have a ton of it, there's really no point in using molecular barcodes. Where it really shines is when you use very tiny amounts of input, such as from single cells or from lower input amounts. So what we've shown that even at 10 nanograms of input cDNA, there is some type of molecular duplication, but when you start putting in 1 nanogram of cDNA or 100 picograms, this roughly translates to about let's say 10 nanograms of total RNA. Then you would start needing to use molecular barcodes, and this would be actually quite important.
Great. Well, I'd really like to thank Billy Lau for this presentation, and thank all of our audience for attending today. We'll leave up this last slide for a few more minutes, and please be on the lookout for other Stanford Inventor webinars, which will be coming up. And that will close our meeting for today. Thanks.